digest reaction. Okay. And then we need our test tubes or our Microfuge tubes to put all the chemicals and whatnot in. So of course, not everybody has this stuff in the comfort of their own home, but you will have this in your lab. And there's two different sizes of Microfuge tubes. I hope you can see them here. This holds about 1.5 milliliters of liquid. This one holds 0.5 milliliters of liquid. Because this device here that I'm going to be using as my heat block only has enough holes for these small tubes, I'm going to be using the small tubes today, but in general, most of the stuff that's done in a molecular biology lab is probably done in these larger 1.5 mil tubes. So I'm just going to get this going. And what we need is a test tube rack, right? We got some place to put these in. So we're going to just line these tubes up in a rack right here, cap them up. I'm going to need 4 tubes to do these reactions. And obviously we want to put a label on each tube so we can remember what each one is. So I'm going to put the first letter of the enzyme that I'm using to do this reaction. That's an E. The enzyme that we'll be using is called EcoR1. I'm going to be putting an H. The enzyme that we're going to be using it's called HindD3. And then I'm going to put an X. The enzyme that we're using is X, M as in mother, N as in Nancy. And then this tube is going to act as my negative control that doesn't have any enzyme in it at all. Don't ask me where these names come from. They're usually about 3 to 5 letters in length. And they don't mean anything as far as I know. I don't know where they got the names from. But once you start using these enzymes over and over again, you kind of know what each one does.
Most of them come from the bacteria that they're extracted from.
Oh, cool. There you go. Learn something new every day. Thank you. So for each of these reactions, I'm going to need DNA, which is the same for everything. I'm going to need buffer, which is the same for everything. My enzymes are going to be different. And then I'm going to put water in each tube to make up the final volume of the reaction that we're doing. And that's going to be the same for everything. So I have 4 test tubes. It's always common practice to make 1 additional test tube's worth of your mix that you want to add to each tube because you don't want to run out. So if I have 4, I'm going to times this by 5. That means I want 10 microliters of DNA, 10 microliters of buffer, 75 microliters of water. What I'm doing here is so I don't have to pipette each thing in each tube. This is a way of saving time where you make what's called a master mix-up. And then once all the common chemicals or common reagents for a particular reaction are in the master mix tube, then you only have to aliquot it once into each tube. All right. So to do this here's what I'm going to do. I'm going to get 75 microliters of water. And here is my micro pipette rack. There's 4 different micro pipettes here on this rack. Each one is able to measure out a certain range of volumes. This one can only measure up to 10 microliters, which is quite small. This one goes from 10 to 20. This one goes from 20 to 200. And this one goes from 100 to 1,000 microliters. All right, so to measure 75, I'm going to need my 200 because 75 is right in the middle between 20 and 200. And to change the volume, I'm not sure if you can see these numbers right here. Let me see what I'm looking at. Oh yeah, this is a little window that has the numbers representing the actual volume that I'm going to be sucking up into these things. You can either turn it here or you can use this black collar right here. So I'm not sure if you can see it. I just set it to 75, which is the volume of water that I want to add into my master mix. This here is just a tray full of these little micro pipette tips that come in these nice handy trays so they stay together, they stay clean, and I could just tip this down, and now I have a tip on the end of my micro pipetteman. And right over here I have my water. So it's always good practice to not put things down if you can help it. So I'm always going to try to hold my top and my my vessel holding my reagent in the same hand. And then it's always good practice to bring this up close to eye level so you can actually see where your tip is, and you can see the water, or in this case, the water or the reagents being sucked up into the tip. Again, this is just good practice that you get used to over and over again. So I'm going to need.
Yeah, Dr. Dave's showing off. This takes you guys literally 3 or 4 lab periods to master. One, just using the micro pipette itself, you get the measurements wrong all the time. We actually do a lab where I have you make the rainbow using different dye colors, and it's pretty easy. Roy G. Biv, and if it doesn't look like Roy G. Biv, you did it wrong. So Dr. Dave's showing off. Like this is a super huge lab skill that he has perfected over years that you guys hopefully eventually will be able to do.
I mean, 20 years, you get decent at stuff, So again, you want to kind of visualize where you're putting your volume of the liquid that you're dispensing. So in this case, I'm just going to put the 75 microliters of water. And then you want to get rid of that tip, because you only want to use a tip once. Try not to use tips over and over again. All right, I'm just going to I cannot say this enough. It's always good to label exactly what you're doing. So I'm going to write MM on this for master mix, so I know that that's my master mix. All right, now I need 10 microliters of DNA and 10 microliters of buffer. So I'm going to switch my micro pipetter and go down to the one that is from 2 to 20. Again, I'm just going to rotate the particular wheel here to adjust the volume to 10. So I know I'm sucking up 10 microliters. I'm going to go get a new tip. All right, I'm going to go get the buffer, which is in this tube here. Again, if something is not in view from my camera, just let me know. I'm going to go ahead and suck up the liquid, suck up the buffer. 10 microliters is not a lot. And I'm going to bring it over here and dispense it into the master mix tube. And then when you want to mix, you can just go like this with your thumb, and it mixes the volume. It mixes the liquid up and down. Shoot it in the waste container. And now I need to get my DNA, which is Lambda DNA in this case. Just a kind of a traditionally used stretch of DNA that's about 40,000 base pairs long. That didn't come up as well. There we go. So 10 microliters of Lambda DNA. Not sure if you can see it. Goes in the tube here.
It looks good. As long as they're pinned, they can see everything. It looked really great.
Awesome. All right. So now I would normally, if I had a pen, I would just check off what I've done so I remember exactly what it is. So I need 15 plus 2 microliters, right, of common reagents that I just made up in the master mix. That's 19 microliters of liquid. So I'm going to stick with my P20 here because it goes up to 20, and I'm going to set it to 19. And now what I want to do is I want to take the liquid from this master mix after I give it a little tap here to mix it up. Just gently tap it. That's enough to mix it. Let's just say if for some reason I'll do it, all your liquid gets spread around the tube. Maybe you dropped it on the floor, and everything is no longer at the bottom. What you need is you just need to pulse it down in this little microfuge. And what this does is it spins the tube really fast, so all the liquid pellets to the bottom. So I'll just do that quickly so you see it. All right, so you can see it spinning, hopefully, and it's going to come to a stop. All right. Why did I put this other tube in here? Because it's spinning so fast, you actually need to balance this out, or else the motor gets all out of whack. All right, so you can now see, hopefully see all the liquid is now once again down at the bottom. And I have a fresh tube on here already. So I just want to take out 19 microliters, put it over there, and put it in each of these tubes. Now, one important thing is to keep track of the tubes that you've already added something into. It's always good to reclose your tubes so dust and your cells and your debris and anything else doesn't fall into them. But I always like to offset the tubes that I've added something into already. So if I get distracted, somebody calls my name, if I have to leave the lab bench, I know exactly where I've left off. So this is just, again, good laboratory practice in a molecular biology setting. So we're going to do this for all of them. Again, this becomes second nature. I could probably fire these tips away with my eyes closed.
That's actually the fun part. And then they start shooting them at one another. And I'm like, wear your goggles, kids. Nobody wants to lose an eye.
So I don't know if I have any P10 tips here, which I would probably go to measure out 1 microliter of enzyme. Instead, I'm just going to drop the P20 down to 1 microliter, and it should be good enough. Now, each of these tubes gets a different enzyme. Everything's been the same so far, right? Water, buffer, and DNA is all you need to do a reaction. And then I want to put a particular enzyme in each of these. So the first one is E for Eco R1. So I'm going to find my Eco R1 tube here. I'm just going to quickly go in and get a 1 microliter is nothing. I don't know if you can see the volume of liquid in this tip, but there's really not a lot there. And I'm just going to add it into. Again, I can offset going up as well. I don't have to offset going down, but this will tell me which ones I've done already, working from left to right. Next one is going to be an H for Hindi 3, which is right here. So each of these enzyme tubes probably cost about $75. You get a decent amount in most of them to use, but they are not cheap. Actually, they're relatively cheap compared to a lot of the other molecular biology reagents you buy, but I don't know who thinks cheap is 100 microliters of liquid for 75 bucks is actually cheap or not. All right, so
it does get crazy.
It does. And then the negative control, I don't want to add an enzyme, so I'm just going to come right back over here and take out another microliter of water. This is my blank, and I'm going to put a microliter of water instead of enzyme into the negative control. In a real world setting, you usually don't do a negative control. I just added it in there, so as good lab practice. So now each of these tubes I'm going to put into this water bath or this heat block, which again can also double as a PCR machine. All right. So I put the tubes in there. I crank down the top because the top has a heated lid to it. And then I need a Chromebook, or an Android device that has the software that runs this machine here. I need to hook this up to the heat block and tell it what to do. So to do that, I'm going to bust out my Android device. Again, you can use a Chromebook if you want to. Plug it in. Hopefully it says no power, which is good, because now I need to turn it on, and it should transfer the protocol to it, the actual program that I'm telling it to. And what I'm telling it to do is I'm only telling it to run at 37 degrees, which is the temperature that the enzyme works best at. So I want to let this incubate at 37 degrees so it cuts the DNA in an appropriate amount of time.
Right, that's what I wanted to jump in. So guys, right now, Dr. Dave took the DNA, he took the restriction enzyme, and restriction enzymes work at an optimal temperature, and that optimal temperature for this lab, for this procedure, is 37 degrees Celsius. So it would actually work all on its own, but it would take longer. So putting it in its optimal temperature allows the enzymes to actually work optimally and do the job the best it's supposed to. So that's what's going on right now.
Right. Worst comes to worst, if you don't have a heat block or a water bath, you can literally just hold the tubes for 20 minutes in your hands, or put them in your pants pocket, which is about 37 degrees from your own body temperature.
Which is what I did in college, because we didn't have all the equipment all the time. So you just walk around. Yes.
So why did I do that first? I did that first because now we have to let this sit for 20 to 30 minutes to let that enzyme work on the DNA and chew it up at a particular site, right? Each enzyme cuts at a different sequence of DNA. So each of those enzymes, in theory, that we added to it, in theory, should be cutting the DNA and providing a different, what we call banding pattern when we look at it on gel electrophoresis. So now what do we need to do? Well, now we need to prepare ourselves for the next step, which is actually looking at our digests using gel electrophoresis. And to do that, we need to make an Agarose gel. I think I told you last time it requires a buffer and it requires some agarose, which is something I showed here. And it literally is just powder that's been purified from agar, and the agar has been purified from the red seaweed. The buffer, I think I said this last time, it's called TBE, tris, boric acid, and EDTA are the initials, or is the acronym. This comes in usually a 20x stock, or 20 times as concentrated as we need it. So we need to dilute this out 1 in 20. And to do that, we're gonna, I've actually, again, this is Rachel Ray. I've already done it here, but what I did is I have 40, let's see if you can see this. Can you see that?
Yep.
Yeah. All right, 40 mils of buffer, or of volume. So really I added 2 mils or 2 milliliters of the 20x concentrated stock solution with 38 milliliters of water. Again, for the sake of time, I'm not going to be doing everything. But then what do you need to do? Well, you need to pour it into usually a glass vessel, and you would either weigh out your particular amount of agarose to make the percentage of agarose in this buffer, weight per volume, whatever it is you desire. Usually we do about 1 to 1.5%. So I'm not going to do the math with you, but you could, as an exercise, I'm sure you might do that. Conveniently, I think I mentioned this last time, this company makes these little tablets, which is basically a pre-measured weight of agarose. And I can just add that in there, swirl it around, and I know I'm getting a particular concentration that I want once it dissolves. So you'll see it looks, hopefully you can see, right? It looks cloudy. It's powdery. The tablet has dissolved into the powder form, and now it's just sitting here in the solution. So in order to dissolve that agarose into the buffer, we need to go down and we need to microwave this to heat it up. How are we doing on time, Mr. Beal? Should I go ahead and show them?
Well, it's 1:20. You got to 1:35, so I don't know how you set up your Rachel Ray.
15 minutes. I think we could probably do it. Let's
little field trip, kids. We're going on a kitchen field trip.
Field trip to my house. You might actually hear or see my daughters. They just finished school. So say hi.
Hello.
Say hi. It's the classroom that I'm teaching to. So here's the microwave. And I'm going to put it in there. And I'm going to set this for about a minute. I'm not going to use the whole time, I don't think. And it's actually important to watch it.
Yeah,
because it will bubble over. Once it starts to heat up, it will bubble over. So I'm going to keep an eye on it.
Especially in that bottleneck. It really loves to blow up in the bottleneck.
You're not going to be able to see it. But after about, I don't know, 25 seconds, I'm going to stop it and just check on it because I'm having a hard time seeing it through the microwave as well. So just pull this open. Nope, it's not boiling yet. Just give it a quick swirl.
Now, guys, I don't know if you noticed it went clear. That means you're getting close. It's just like making Jello. You want that cloudy liquid to go clear.
Yep. I'm going to open it, and you're probably going to see it. Oh, not bubbling yet. All right. Let's give it another quick swirl. I'm trying to wait till it boils, and then I'm going to open it up so you can see it kind of boiling over. Not boiling over, but boiling up the side, which is. Did you see it boiling?
A little bit. Yep.
Can you see it bubbling in there? I'm going to turn it on one more time to make sure it's all dissolved. And you might think this is funny, I'm doing this at home, but this is literally how I would do it in the lab every day.
Yep.
Just got to keep an eye on it, make sure it doesn't boil over. And now hopefully you can see there's no more powder in there. Everything is dissolved.
Super clear.
Liquid.
And this is everything Dr. Dave is doing is what you guys would do if we were not virtual. This is all the same techniques that you would be doing.
All right. So now obviously it's hot. I'm using a paper towel to not burn my hands. Walk back upstairs. Now we need to add a dye to the gel. Usually we're going to wait till it cools down a little bit, but I'm going to do it now for the sake of time. We need something that's going to bind to the DNA and fluoresce so we can see it when we do the gel electrophoresis. Without this dye, your DNA will be there. You just won't be able to see it. We used to use some really nasty stuff called ethidium bromide, which is cancer causing. I think I might have mentioned that last time. Now we can use this stuff called gel green. And this is concentrated 10,000 times. So I don't have to add a lot. To the 40 mils here, 10,000 times, I'm going to add 4 microliters, excuse me, of this stuff, which isn't a lot. It is light sensitive, so which is why it's kept in a black bag.
And guys, that's important. Anything light sensitive comes in a brown bottle or a black bag in a lab. Like that's right away. If you see that, you're like, ooh, can't keep that in the light.
No, I mean, I could keep it out for the next 20 minutes if I had to, but I just like to be safe and do that. And now hopefully you can see, let's see, it has a little red tint to it, a little pink orangey tint to it. That's the dye that's in the gel now, that when the DNA runs through this gel, it will bind.
Awesome.
Right? So here is the casting apparatus. It has a little insert, which is where we're going to pour our gel. It sits in there. And then I think I mentioned last time you need a comb, which creates the wells in which you'll put your DNA. So really, it's all put together here nicely. And what I'm going to do is I'm just simply going to pour the molten agarose into this. It's all the way up to the top. I probably should have poured a little bit less, but that's all right. And then we're going to let this harden or solidify back into like a jello type of substance. Again, so I don't have to wait for that. I've already prepared this gel. It's like pulling your dish out of the oven and on a cooking show. So here it is. Right, I pull out the comb. You got to pull it straight up, and then hopefully, let's see, what does it look like on my screen? Hopefully you can see the wells. Yeah, it looks like
we can see it.
All right. So each of those wells holds probably about, I'd probably say 40 microliters or less of liquid. Our reaction volume, if you remember from the recipe, is 20 microliters. So we can use that. And now the fun part is actually loading your gel. In theory, we would have let this sit a little while longer, the reaction. We're probably going to stop it and we're going to pull it out. And I'm going to use the precast gel that I've already made here. And I'm going to put it in the gel loading chamber here. And what I should have done is I should have made more buffer, but for the time being, so I should be, if I use buffer in the gel, I'm going to be using buffer in my gel electrophoresis chamber. But for now, I'm just going to fill this up with water so you can have an understanding of what we're going to be doing. You want to cover the entire gel with your buffer. So remember, it's electricity, basically the current, which pushes the DNA from the wells into this gel matrix that we just made and allows the DNA to separate by size depending on the concentration of agarose in your gel. All right, so here I'm just going to pause this reaction now, and I'm going to open it up, get my cube rack here, take these bad boys out. And one more thing we need to add to this. It serves two purposes. We need to add what's called a gel loading dye. It's usually purple, which is right here. And it usually comes in a 6 times concentration. So we got to get this down to 1x. I have 20 microliters in each of these. If I add 4 microliters of this, the total volume now comes up to 24. And if you divide, if I use 4 microliters in a 24 microliter reaction, the math comes out to make it 1x, trust me. All right.
A lot of math in genetics, guys. A lot of like basic, simple math, lots of percentages. So, you know, don't be intimidated.
Don't be intimidated. You start doing the stuff in your head after a while. So I need 4 microliters, which is what my P20 is already set on. So I'm just going to now take this out. I mean, take off the top again with one hand, the top back on, and I'm just going to be adding this dye to it. This dye serves multiple purposes. The first thing it does, it stops the reaction from continuing. So I just blunted the enzymatic reaction by adding this dye. Second thing it does, it gives you a visual indication of how far your DNA is running through the gel. With the purple dye, you can see it directly within the gel. And then the final thing it does is it contains glycerin, which is heavier than water and heavier than the buffers. So as you mix that into your reaction, when you load your DNA into the wells, you want your sample to fall to the bottom of that well. Or else if it's the same density as water or the buffer, it could just float up and you would lose your sample. So you want to make it slightly heavier than the buffer that's in the chamber. So this glycerol actually does that, which allows the sample to fall to the bottom of the well. Again, I'm offsetting my tubes. I cannot say that enough. It's critically important. So you remember what you're doing. Sometimes you make up reactions that have, 10, 15 different parts in each of them. And you got to make sure you remembered which ones you added to which tubes. So now, reaction stopped. We have 24 microliters of liquid in there. So I'm going to grab my P200, which goes from 20 to 200. So 24 is in that range. And now we want to load it into our gel that's sitting right here. To do that, you're just going to open it up. You're going to take out your entire sample. I'm not sure. Hopefully you can see it being sucked up. All right, now the purple stuff's in my tube.
Yep, we can see it.
It's hard if you're going to do this with one hand. Look how much my hand is shaking. My hand has never been that steady. So to get it in that well is slightly difficult. So a technique that I've always learned is to just rest this side of my hand on the base of this, and then just touch this with one of my fingers, which helps to stabilize it as I'm loading my gel. You also don't want, you want to put your tip of your pipette min directly into the well, just under the edge of the well, or just under the edge of the buffer level. You don't want to jam this thing all the way to the bottom of the well, because then you're going to puncture a hole in your agarose and your sample will bleed right out the bottom and you won't be able to see anything. All right, so I'm just going to choose the second one in. You can see hopefully now that can you see the purple liquid sitting in the well?
I always have the kids use their cell phones too. So I have the cell phone light on behind it, and that light illuminates and it makes it a little easier for them to see the wells, because all my physics nerds out there are like, objects look different in air than they do in water. So the refraction, it almost looks like the pipette min is bending when you put it in water. So it makes it even more difficult to load the well. Yep, we can see the purple. It looks great.
So I'm comfortable doing all of this stuff without any gloves on, because I'm really not using anything that's going to be harmful to me. It's DNA. I have DNA. It's buffer. I don't have to worry about the buffer. It's just some salts. I'm not using that really nasty DNA intercalating agent called ethylium bromide again, which is what one would normally use. So I'm totally comfortable using without gloves. But your teachers and your lab colleagues might encourage you to use gloves. And most oftentimes I did, because I don't want any of the enzymes or anything like that from my own hands getting into my reaction or onto my samples. But for today's purposes, I'm just pointing out that I don't have gloves on. So then this thing is pretty simple. It's pretty foolproof. You can't put it the wrong way. Remember, you want your DNA to flow, your current to flow this way. So I can't do it like this. If you did it like this and the current is flowing this way, I would run my samples right up out of the gel through the top of the gel, and I would lose them. But it doesn't allow me to put it on that way based on how it's made. So the only way I can put it is this way, and the current's going to go this way. And all I have to do is push Whoops, I didn't plug that one in. All I have to do is push power button. This one is plugged in, so you can see the green light comes on. What you'll notice is at the metal electrodes, which is creating the current in the chamber, you will start to see bubbles very quickly coming off of the metal electrodes, and that's a good sign that you now have current flowing through your chamber. And then, like I said, this thing is just like a little. Oops, let me clean this off. Turn off the power. This does get foggy sometimes.
So
so I'm just gonna.
When you run the current through, guys, it separates the water into hydrogen and oxygen gas, and the water, because you're running a current through, it gets warm, so it creates a lot of condensation. So it's difficult to see the DNA after about a half hour during the run.
And the only other button on this chamber here is the light source, and this is a blue light which will illuminate that gel green dye that we put into our gel in order to visualize our DNA. So if I push this button, you'll hopefully saw a blue light come on down here.
Yep.
And then the only other thing, oh yeah, it's on my head. The only other thing I need is my cell phone to actually document what we're going to be looking at here. So I'll flip my phone around so we can actually see the camera. And
there it is.
This is the gel that I ran earlier today. I can't take a picture of it right now because I'm not in my, so I don't know what that looks like. What does that look like to
Looks
you guys?
good. You see, did you use the ladder? I can't remember.
I did. You're right. I did not mention that. I did add the ladder to this gel that I ran before. So in order to tell, right, you're going to see a bunch of bands after you do a digest of your DNA. Your enzymes are going to cut the DNA into a particular sizes, which will then run through your Agarose gel and get caught up at particular size ranges in your gel. And this can be pinpointed down to about 50 to 100 base pairs. So you would normally see multiple bands, and you need this ladder on the left-hand side. This is a standard ladder sizes. Every band is about 1 kilobase away from the other one. So you can, this probably goes from the first one you can see there at the bottom is 2, then that slightly brighter one up from that is 3, and then it goes 4, 5, 6, and then they kind of all start bunching together because of the percentage of gel that I made for this particular experiment.
What Dr. Dave is saying there, guys, is each one of those bands is 1,000 base pairs, basically, right? So it's 1,000 ATGs and Cs. So like the joke I always say, because it's football season, the really big chunks of DNA are your linemen, and they move slower through the gel. So they get closer, they stay closer to the wells compared to your wide receiver and running backs that run quickly through the gel. So your small pieces of DNA, your small BPs are in the 1, 2, and 3,000s, and your large pieces of DNA are those 4, 5, and 6s. So that ladder you see on the left, it's not a control, but it lets like people like Dr. Dave know that the chamber worked and you got to run and it looks decent. And then everything to the right are your experimental DNA, the experimental DNA that you digested using your restriction enzymes.
And when you're, oftentimes when you're creating a piece of DNA that you want to put in a foreign organism, you are basically using these enzymes as scissors, cutting out portions of DNA from sample 1 and putting it into DNA from sample 2. It's literally like cutting and pasting with scissors and glue. And in order to make sure that you have what you think you were going after when you were originally setting up this craft, this DNA craft, if you will, cutting and gluing, you could sequence it. But the old traditional way, and I think a lot of people still use this way, is to simply do a quick restriction digest and make sure that the size of your bands are in the range that you think they should be. Maybe if I did a digest, I would get a 1,000 base pair band, a 5,000 base pair band, and an 8,000 base pair band. And if I see that on my gel, then I know I'm probably pretty good and I can move on to the next step. You can see actually, and I took a look at this gel earlier today, this reaction, and I don't have a good reason why, this reaction did not work. you can see that the ladder looks great, in my opinion. You can see multiple bands of different sizes on the left-hand side, but you just see this big clump of DNA at the top towards the top of the ladder. It's run into, that lineman has gotten into the gel a little bit, but not very far, if you can look at the sizes of the ladder. So I don't have a great explanation for you why this didn't work. I store these enzymes in my home office freezer. One of these little, like, whoops, let me just flip this around. One of these little fridge freezer units. Maybe the freezer part didn't keep them as cold as they should have been kept, right? These enzymes need to be kept pretty cold. Maybe the freezer itself, unbeknownst to me, kind of freezes and thaws during its normal operation. So as it's thawing, my enzymes actually went to 4 degrees instead of staying at -20. So maybe the enzymes are actually bad at this point. I can't really think of another way. Another reason, maybe my buffers went bad.
That happens. It happens all the time, guys. Like,
yeah,
every third
this is
lab
science,
we do in
right?
here is a success. It's like, I tell you guys, it's like baseball. If you're 1 for 3, my dad said it growing up, if you're 1 for 3 in baseball, you're a millionaire. Same thing with genetics. Like,
same thing.
Every third lab works and the other 2 sometimes don't, and you start over.
And I could have made up a story and said, oh, look, this looks great. But I didn't want to do that. I wanted to point out that even something as standard as a restriction digest sometimes just doesn't work. And then you need to go back and troubleshoot why, or just redo it and figure out what happened. So this is science. And this is one of the reasons why I wanted to show it to you. Something that I thought was going to work. I could have done this with my eyes closed in the lab, but it didn't work this morning when I set up these reactions. Maybe my heat block actually didn't go to 37 and it sat at, 4. I mean, it sat here at room temperature, which didn't allow the reaction to proceed as efficiently as it should. As your teacher said, it should still digest. It'll just be slower. So I really don't have a great answer.
It was great. So Dr. Dave, they created a spreadsheet full of questions. Guys, I'd like you to circle back to that spreadsheet and add some questions to it based off of today. I know you guys have to go to your next period. What do you guys say to Dr. Dave?
Thank you.
You're welcome.
Hope
you learned something. Hope it was good.
That was great. It really was great. All right, guys, you guys have a good weekend. I'll see you next week. And like I said, flex period, go back to that survey and add some questions, because I'm going to forward all those questions to Dr. Dave after the weekend so he can respond to you guys.
Cool.
Okay.
Thank you.
Have a good one. Thanks.
Bye-bye.